So thanks for that introduction. I'm excited to talk about anomaly detection in SU data. So it's a high-level motivation. One of the most fundamental questions you can ask in tissue biology is trying to understand how tissues that undergo some perturbation, whether that be disease or injury, are somehow different from some healthy reference condition tissue. It's only been recently that this class experiment has been attempted with spatial transcriptomics data. And so how this might be set up is that you have samples that are healthy or diseased tissues, and you collect one or several slices from each of these tissues. What you're given at the end of this experimental pipeline is a spots by gene expression matrix, as well as a 2D coordinate for each of these spots. And you'd have this pair of matrices for each condition in your data. So given this data, one motivating question you might want to ask is, what are the gene expression and location pairs that are associated with condition change? Or in other words, what are the anomalous cell states? So this question has been posed in similar forms for expression data and for location data independently. If you cross out location, just think about expression, there are a large number of methods in the single cell literature that define anomalous regions on the gene expression manifold as regions in the manifold where one condition is differentially abundant. If you cross out gene expression, think about just location, these anomalies are classically defined by spatial scan statistics. However, in this sense, there's no existing method that formalizes these anomalies for high-dimensional gene expression and spatial data. So our contribution is twofold. We first formulate this spatial differential abundance region problem, or SDARP, that defines what these anomalous cell states are in SU data. Secondly, we present Sardine, which is our framework to solve this SDARP problem that sort of neatly combines ideas from both scan statistics and these single cell differential abundance ideas. So focusing first on this first contribution, it might be easier to set up this problem by just considering gene expression first. So in this setting, we're once again given healthy tissue and diseased tissue, but instead of spatial measurements, we're just now just given dissociated cells, which are represented by these orange and green cartoon cells here. Now we assume that there's some condition-specific density over gene expression space that each of these cells draws its distributions from, or gene expression measurements from. And so for the healthy cells, they're drawn from one distribution, and the disease cells are drawn from a different distribution. So we assume that this gene expression values live on some low-dimensional manifold, and without loss of generality, we define some anomalous region as the region on the manifold where the perturbed density or disease density dominates the healthy condition density. So in this sense, to find the anomalous region might be easy if you had access to these densities themselves, but in reality, we don't have access to these two densities. We do have access to is just a finite number of observations. And so this motivates the single cell differential abundance problem, which is that for these two densities sport on the same manifold M, we're given observations x and x prime, which are these two expression matrices. And we want to find all the points in our data that belong to this anomalous region. And as alluded to before, variations of this problem have been solved for a number of different methods over the past number of years. But it might be obvious to many of you in this room, but this is not sufficient for thinking about spatial data. So it's a very simple example. We thought about two tissues in two different conditions, healthy and perturbed, where perhaps you're interested in just one gene that increases in expression from left to right, another gene that increases in expression from right to left in the other condition. It's entirely possible that these two could have the same gene expression densities, yet clearly you want something interesting about why these two tissues are different. So the problem is that S0 and F1 do not include spatial context. But we can fix this by first considering a particular region on the common coordinate system. And I'll make a small technical note here that we assume that the slices are already aligned or pre-registered before they're given to us. A number of tools have already been designed to accomplish this pre-processing step. But for a particular spatial region R, we can find two conditional densities that are the gene expression densities, given that the spot is observed within the spatial region R. And for a small enough window size, this approximates the pointwise conditional density for a particular spot or a location of a spot on the tissue. So this motivates the spatial differential abundance region problem, which is sort of a natural extension of the single cell version of this problem, where now we're given not only the expression matrices X and X prime, which are defined in the same manner as in the single cell case, but also S and S prime, which are the spatial coordinate matrices. And so given a spatial window R for each spot, we want to find these conditional densities for all the data points in our data. So our second contribution is Sardine, which is our method to actually solve this problem. This is an overview figure, but I'll break it down into two simpler components. We first define a set of spatial neighborhoods, which we call the spatial anomaly family. And it's going to be flexibly defined as a function of each spot S. But for the results I'll show, we default to the construction of the K near spatial neighbors in each condition to each spot S. And secondly, we require a plug-in density estimator. There are also a numerous number of choices you can make for this, but I'll present a reticular implementation that I think is well suited for this problem. So in prior literature, there is this idea of density estimation on the manifold graph. So given some observed gene expression measurements from both conditions, prior work both in stats and ML, as well as in the single cell literature itself by Burkhardt et al. In 2021, proposed this idea where you can just estimate the density values at the observed data points. And the way they do this is by first constructing this joint manifold graph from both conditions simultaneously. And this is a common step in a lot of single cell pipelines. And the way you get density estimates is by first, for one condition at a time, assigning uniform probability mass to all the observed data points in that condition, creates a signal vector D. Next, you can compute density estimates D star as a solution to a reconstruction problem with a Plotchman regularization. And so you balance finding a D star that's close to D, but also is smooth with respect to the edges of this graph. Sardine extends this naturally to the spatial setting by first having the first initial step of the solid-state manifold graph from the joint union of the observed expression values. But when you create these signal vectors, you only do so for the observed data points within one particular spatial region at a time. In this setting, the resulting density estimates you get once after smoothing are spatially localized, that particular spatial region that you assign the probability mass initially at. So finally, the output that we show is the likelihood of being perturbed for a particular spot, and we average this number across all perturbed spots within a particular spatial window. And we do this across all spatial windows in a particular tissue. And we'll contrast this or benchmark against two other recent methods, Stans and Vespucci, which are these two screenshots on the right side, which are heuristics based on classifier accuracy. And we'll also benchmark against MELD, which is by Burkhardt 2021, which is one state-of-the-art single-cell RNA-seq differential abundance method. So to set up some simulated results, we constructed the following synthetic case, where we simulate some manifold structured data, which is shown on that left, as well as two spatial slices, which are these two checkerboard boxes nearly to the right of it. In each checkerboard square, we have 11 by 11 or 121 individual spots that are each sampled from the same location or region of the manifold. So in this example, if you look at these two artificial slices in the top right checkerboard square is purple and blue, that means that for the first slice, we've sampled all of those spots from the purple region on the manifold. And on the bottom slice, we sampled all those points from the blue region on the manifold. And so comparing these two slices together, all the locations or checkerboard squares where the location on the manifold has changed are what we call the anomalous squares. And I guess on this screen, it's a little bit faded, but there's like an L, backwards L shape on the right side, which are the anomalous squares. So comparing against both MELD and Vespucci, I think both quantitatively and qualitatively, we recapitulate this known ground truth the best. The other method, Stans, that I mentioned, requires histology data as an input, and so we didn't benchmark it against it for the simulated case. On real data, or on real biological data, we looked at a data set from Corporgitu et al., Nature Communications 2024, which studies the mouse cortex injury response by 10x Visium slices of healthy versus injured slices. And so this figure, which is taken from their publication, we have a healthy tissue on the top, and then a injured tissue on the bottom, where this dotted line here indicates an injury that they've induced into the tissue. And so first, we preprocess these two spatial slices by aligning them to a common coordinate system using ST-align, which is a recent method by Clifton et al. In Intercommunications. And then we ran our method. And the result is that Sardine most closely matches the known injury site tissue geometry. And so our result on the right, and the left side, bottom, is where that stab wound occurred. And Sardine has the most, I guess qualitatively, you might imagine that it matches the shape of the stab wound the most closely. And also we observe that the anomalous region folds into that cortex region, which is also a phenomenon that was observed in the original manuscript. Furthermore, if we assume that spatial coherence is a good indication of the result quality, Sardine also has the high spatial coherence as measured by Moran's eye. Finally, Sardine's anomaly scores have a higher correlation with known injury response genes. And so again, from that original publication, there were a number of marker genes for particular cell types, as well as this type of interferon pathway that was shown to be positively correlated with the injury site location. By comparing our anomaly scores for each method, the correlation to these marker genes, we show that Sardine is either has the highest correlation or tied for the highest correlation across these different categories. So to summarize, we formulate the spatial differential abundance region problem and propose Sardine as our solution to it. And this mouse cortex data set, as well as one other data set that I didn't show in this presentation, we discover highly plausible perturbed regions. As future work, we plan to benchmark on different data sets, different spatial resolutions. Since both of the data sets in our manuscript are from Visium, it'd be interesting to try on data sets of high resolution. And furthermore, another interesting question one might ask is this initial preprocessing step of alignment typically assumes some sort of correspondence in expression space between different slices. And so it'd be interesting to solve an anomaly detection problem alongside an alignment problem together. But with that, I'd like to acknowledge my advisor, Ben Raphael, and my collaborators, Utsav, Julian, and Sune. I'll take any questions now.
Thank you, Gary. Very interesting work. Are there questions? Yes.
Good talk. Thank you, first of all. Why did you define anomaly by increase of the density of, it could be also decrease, right?
That's true. You can define it either way if you wanted to. Or you can flip the, we consider the healthy slice and the perturbed slice, however you define it.
Or maybe define a region around the same density where it's similar, and then consider the other sides, like a volcano plot.
I guess there are many definitions of anomaly, I guess, in the literature. Historically, this is one that's been used a lot.
Thanks so much. So differential abundance analysis is totally popular in single cell, I agree, and could be up, down, whatever, it doesn't matter. But I think spatially what you do, I'm not totally sold that you can really assume that you have sort of full alignment of tissues. You know, often, you know, we have to say niches or something like that are sort of co-consistent, but let's say you have like a bad tumor or whatever, right? These things just cannot be aligned across different samples. So I would encourage you, and maybe you must have thought about this, like how do you deal with the fact when like pre-alignment, which is sort of a major assumption for you to have sort of XY overlapping when this one does not work?
Sure. I have a couple of answers, I guess, to this question. It's a very good question. I guess the first is that there are many, I guess, tissue systems that are alignable, and there's been a success with existing. What? Yeah, sure. But I guess it's an open question of how you'd apply this method when that isn't the case. I guess one thing to think about maybe is that when tissues aren't exactly perfectly alignable, the problem setting itself is kind of ill-defined or hard to interpret. And so I guess our tool was designed for tissue systems that were, I guess, easily aligned. You'd require probably a very different setup entirely for when you can't assume like a common coordinate system to begin with. Sorry, can you say that one more time? Sure. So you're just, if I were to interpret your question another way, you have a lot of common corner frames with just small regions as opposed to one common corner frame across the entire tissue. Right. That's a good point.
Yeah, thanks a lot for the talk. So I think in the last slide you were showing, you were alluding to how, you could correlate the, the anomaly score you had with, this expression of specific markers. But in practice, do you find you could distinguish cases where the anomaly had to do with a shift in, say, the cell type distribution in that region, so as opposed to just a change in a specific gene program that could be underlying a cell type that's there?
Right. I guess one advantage of our method is that it doesn't require cell type annotations to begin with. And I guess there's also some argument to be made that defining these anomalies strictly in terms of the gene values themselves, as opposed to these discrete labels, gives you better resolution or more information to begin with. It could be entirely possible that cell types themselves change the distribution between different conditions. That'd be interesting to find through this method as well.
Oh, we have an online question, and since they do not have the opportunity to ask questions elsewhere, they will get a preference.
Right. Ian Lee wishes to know, does the algorithm consider different cell type at different location?
So I guess our algorithm is like cell type agnostic. We don't like require annotations of cell types, nor do we like label cells or spots into cell type labels.
Okay, and do we have a comment more rather than question from Jose Lugo Martinez? As a schematic comparison as a function of alignment quality would be really cool.
Sounds good.
She has one more last quick question.
Thanks. Going back maybe to this idea of the fact that you probably cannot get a global alignment and you might have a local alignment, maybe what change your framework to consider the statistical properties of the tissue in a way that is not really requiring alignment. I wonder if you can generalize your framework, because in fact, what you're looking for is the match between the gene program and the distribution in space. So this might be a more global feature than just the definition of local anomalies, right? Do you think it could be done?
I think there's a lot of interesting work to be done on the case where the slices can't be aligned, that you require a different setup maybe than what we had here.
Okay, with that, we thank you again.